Assalamu alaikum. Welcome to Darwinian Delusions. And with me today is somebody who definitely doesn't need an introduction. If you are in tune with what's been happening in terms of evolutionary thinking for the past couple of years, then I'm sure you would know of Dr. Paul Nelson, who is a philosopher of biology. And I don't want to get into the long introduction I did last time, simply because we are short on time. How are you doing, Doctor?
I'm doing wonderfully. Thank you.
So it's wonderful to have you back. I had a lot of very positive feedback from the last live stream. In fact, surprisingly, the stream was doing a lot better after it was online, and even weeks and months later, comments and questions and whatnot. So there's a lot of demand for you to come back. So welcome back. So we're going to go for an hour today. Topic of orphan genes, of course, and this is something which I may just bring you back maybe another time just to complete it. So let's just get into the thick of it. Orphan genes, I'm just going to give you a very brief overview, everybody, is a topic which I'm proud to say, and I did say on my last live stream, that I'm blessed to be speaking to the person who went against the grain and made some risky predictions, which were confirmed. And those predictions, I would say, if they were not confirmed, would be pretty much career ending. So Paul took quite a bit of risk making those predictions. So over to you, Paul. Tell us about orphan genes.
Wonderful. So I'm hitting my share button here.
Sure. Sorry, that's my WhatsApp. So for anybody who is interested, just as, here the slides already.
Yep.
I'll be quiet. Here we go. Over to you.
Okay, so this topic, and thanks for having me. I really enjoyed the last time I was with this group and look forward to future discussions. The topic of orphan genes has grown so rapidly over the past decade that today I can really only set the stage for the discussion. And on February 15, that Monday, I will be doing part 2 with another group from the UK. I've given the link there in the title slide, and I'll also provide the YouTube link to Subor. Is it Subor or Subor?
Both work.
Oh, okay. In part 2, I'll be dealing with proposed evolutionary mechanisms for the origin of orphans. So the topic is very rich and full of fascinating details. This you can consider sort of laying the foundation or setting the stage. Before I get to that, though, let me give you a heads up on a very interesting talk that is coming up on February 11. So that's this upcoming Thursday by Nathan Lentz from John Jay College of the City University of New York. He will be in San Francisco talking about his research on human orphans. So he has discovered apparently hundreds of human-specific microRNAs in our genome, and he'll be presenting this research, which has been going on for some time, in this presentation. The link will be available online, so you might want to make a note. It's not available yet. Make a note if you're interested about human unique orphan sequences. Dr. Lentz will be talking about them. And I should make sure I don't misrepresent him. He is not, like me, an advocate of intelligent design. In fact, he strongly opposes that view. But I can't wait. I know he's been working on this, as I said, for quite a while. And this is, I think, the first public statement of what he's found. So I'm going to be tuning in. I hope to see you there. All right, let me give you a little anecdote that's quite revealing with respect to the topic we're going to discuss today. In April 2019, I was at Churchill College in Cambridge for this meeting, a wonderful meeting, by the way, dealing with the so-called extended evolutionary synthesis. And I was presenting a poster there with Professor Change Tan of the University of Missouri, actually a poster on orphan genes that are apparently essential in different groups. And let me say, by the way, if anyone is interested in this PowerPoint file, I'm happy to make it available to you. You can use it for your own research or study. And my only request is that you not put the PowerPoint up on the web, that you use it just for your own study. But if you want any of these slides or the whole presentation, contact me, care of Discovery Institute, and the staff there will forward your email to me, and I'll send you the PowerPoint file. So feel free, if you want the file and you don't want to take notes now, I'm happy to send it to you. So anyway, I'm at this meeting, and really a very interesting event. And I was at lunch one day with a young postdoc whom I won't name to protect him, but we were sitting in the dining hall of Churchill College, and there are hundreds of evolutionary biologists around us from all over the world. And the topic of universal common descent came up, the great tree of life, Darwin's, I would argue, his most significant theoretical contribution to science was really not natural selection, but rather this unifying geometry of all life being linked in one universal common ancestor. Anyway, that topic came up, and Professor Tan and I are both skeptics of universal common descent. So we were talking about it, and he kind of hushed us, and he said, "Look," and he's looking up and down this long table in the dining hall, and over his shoulder, he said, "If I challenged universal common descent, audibly, loudly enough that other people could hear me," so we're sort of speaking in whispers across the table, he said, "I would bring every conversation in this room to dead silence." Now, he knew, and we knew, that he was right. And he went on to say that that wouldn't be the case with certain proposals of teleological evolution, such as advanced by the Cambridge University paleontologist Simon Conway Morris. So it was clear at that moment in April 2019 that there were relative degrees of certainty within evolutionary theory about universal common descent. And it was, in a sense, the holy of holies. It was the geometry or the topology of historical relationship that it would be dangerous, literally dangerous to one's career to challenge. So we need to keep that in mind. We need to have a certain realism about the centrality of this theory to current evolutionary thinking or biological thinking in general. But I think things are changing. As I'll discuss in a few minutes, I think the genomic evidence is pressing very hard against this geometry. And what I wanted to say to that young postdoc, but didn't have a chance that day, was the matrix of opinion about this question is actually well populated. So here on the left, we have LUCA, the last universal common ancestor, existed, and two alternative views about the causal mechanisms involved in building the tree of life. In one, there's no design. In the other, there is design. So these will be two figures that are sort of characteristic of that viewpoint: Charles Darwin himself and Stephen Jay Gould, the late Stephen Jay Gould, whom I got to know a little bit when I was a graduate student. Now, on the right, we have the non-existence of LUCA. There was no entity that we could say was the ancestor to all life on this planet. And if we take the familiar quadrant of this matrix, that would be creationism. So we have Linnaeus there, Louis Agassiz, the last great American creationist at Harvard University in the mid to late 19th century. So typically when people think about this question, they diagnose it between these two poles of opinion. But in fact, every quadrant in this matrix is populated. So there are design theorists like my friend Michael Behe, or the 19th century correspondent of Darwin at Harvard, Asa Gray, the great botanist, who think that there was a tree of life, but that the tree of life only came about because of design. Now, the perhaps unfamiliar quadrant is this one. LUCA did not exist. There is no tree of life in Darwin's sense, but there's also no design. So here I've placed the late Carl Woese, whom I met as a graduate student in 1988 at Woods Hole Marine Biological Laboratory, who did path-breaking work on ribosomal RNA to give us the three-domain picture that I'll show you in a moment of the tree of life. And then still very active and really a very philosophically oriented biologist. I love to read his work. W. Ford Doolittle at Dalhousie in Canada. Now that fourth quadrant there in the upper right-hand corner is becoming increasingly populated, as I'll show you in a minute. And my point in showing you this matrix is to realize that the diversity of opinion about this question is actually much greater than people understand. And it helps to lower the temperature in the room to realize you can migrate conceptually among all of these quadrants, learning something from each of them. I certainly have done that. So, quick outline where we're going. I want to lay the analytical framework that Darwin himself and his intellectual offspring in the 20th century saw as fundamentally testing common descent, and that's something I'm going to call the principle of continuity. Secondly, we'll look at some lines of evidence that may violate the principle of continuity and hence by the theory's own standards falsify common descent, and that's where the orphan genes will come in. So in part 2, which I'll present on the 15th, I'm going to look at a response to the second point of this outline on the part of evolutionary theorists who say, in fact, we have mechanisms that can generate apparently de novo coding sequences, protein coding sequences, and therefore the pressure on common descent provided by orphan genes can be relieved. Okay, so I'll go into much more detail in the second part about those evolutionary mechanisms. But here's the scientific universe in which we find ourselves. And like so much else in biology, it owes its outlines, its shape to Darwin. So in his great summing up in chapter 14 in The Origin, and this is from the first edition, he says, the affinities of all the beings of the same class have sometimes been represented by a great tree. So he's extending this argument of affinity from trees, let's say, for the vertebrates or for the mammals to the whole of life. He says all living things have much in common. Therefore, I should infer from analogy that all the organic beings which have ever lived have descended from some one primordial form into which life was first breathed. Okay, so we can take these notions present there in 1859 and map them congruently onto the concepts that we have today. So for affinities, read molecular similarities in DNA, RNA, proteins, a great tree, universal common descent, and some one primordial form, the last universal common ancestor. So in outline, the theoretical landscape that we're considering is very much set in place for us by Darwin. Now, it's hard to represent the even a tiny portion of the full diversity of the tree of life in a single diagram. So often what's done is a kind of rolled up form or a mandala representation like this, where the tree, instead of extending horizontally, is rolled up around LUCA. So you can see here we have the three domains of life, the archaea, the bacteria, and the eukaryotes. Metazoa is over here, this very large group over here. We're on one of these branches. I'm not sure which one. The amount of detail here is bewildering, but the thing you need to keep in mind as centrally important is the existence or non-existence of LUCA. LUCA will be the key player in our story because its existence creates a biological singularity or establishes a theoretical singularity that strongly constrains what we may infer about the subsequent history of life. So if LUCA existed or LUCA did not exist, make a major difference. In fact, I think it's safe to say it makes the most important difference to how you do historical biology, how you build phylogenies, what molecular similarity, molecular homology means, what the rules of inference are for constructing phylogenies, and so forth. If LUCA did not exist, then at a fundamental level, everything in historical biology changes. Now, I don't have time to go into this in the detail today that it deserves, but methods of phylogenetic inference change radically if LUCA did not exist. If you had multiple independent starting points, let's say for the ribosome, the molecular factory that builds proteins in all cells that we know, if you have multiple independent starting points for that entity, the meaning, the historical significance of a ribosomal RNA changes radically. Anyway, I won't belabor the point, but keep that in mind. LUCA matters. So here's just back up a sec. This is typically how the tree is rendered. Now, this doesn't look like a tree, right? But you can imagine that it's a tree if you picture yourself floating above it, let's say in a hot air balloon, and you're looking down on a great oak tree or a big maple, some tree with three major branches. The bacteria and the archaea are the single-celled organisms lacking nuclei. Here are the eukaryotes that have nuclei. We are on this branch here. And of course, there's a great deal more diversity to eukarya than I think most biologists even typically realize. But LUCA is located here at the origin, where this tree goes into the prebiotic soil. Theoretically, the important thing about that three-domain picture is its universality. The scope or domain of the theory is universal with respect to life on this planet. There's nothing outside it. In terms of the logic of biology, that turns out to be very important, as you'll see. So LUCA's here, and what you need to remember about LUCA and all of its offspring, including you, right? And the E. coli in your gut, and my cats were in here marching around just a moment ago, my cats and so forth. The universality of that scope is constrained by the fact that evolution is not unconstrained. I'm trying to put that more simply. What you don't want to happen as an evolutionary theorist is say that anything can happen, because your theory loses all predictive power in a case like that, so that it's consistent with whatever you observe. That is not a good theory. That turns, excuse me, one sec.
Yeah, and just to add to Paul's point there, basically make homology and homoplasy just irrelevant. Everything would be just up in the air.
Exactly. Everything would collapse into it. These concepts would collapse into each other, and they would end up being useless to you. So there's some unknown prebiotic pathway, let's say, to LUCA. The first cell gets up and running. LUCA is going to be a very large critter, right? It won't be stripped down like a virus. It's going to need a ribosome. It's going to need dozens of very large RNAs to build not only the ribosome, but some of the other key molecular machines in the cell. And because of functional necessity, what works in LUCA is going to be retained by its offspring. So as you climb up into the tree of life, or conversely, as you climb back down to LUCA to infer its characteristics, recognize that evolution is constrained. Not anything can happen. Evolution's often been compared to taking an airplane off at an airport, let's say a little Piper Cub or a little Cessna, and while the plane is flying, you rebuild the engine in the air, right? What can't happen is a crash. You've got to keep the plane aloft. In a case like this, where we take that kind of analogy back into biology, there's no dying in evolution. So viability strongly constrains what you're allowed to infer as you work your way along the branches of any phylogeny. All right, keep that in mind. This is vitally important. Evolution is not an unconstrained process. So when we come to key features of LUCA, or that we infer must have been present in LUCA, our ability to form hypotheses is going to be constrained by these viability considerations. And this is, in fact, what informs the principle of continuity. So another way to think about this is you need to keep your eye on the deeper logic. This rarely, if ever, happens in biology, where you go straight from common descent to predictions about observations. In a logical schema, what's much more common, in fact, I would say it's universally the case, is that common descent, excuse me, common descent is coupled with auxiliary theories from other parts of biology, and that lets you make predictions that you can then go out and check. So those auxiliary theories give you specificity with respect to, let's say, the genetic code. So here's an example using the genetic code. The 64 trinucleotide translation table between the realm of nucleic acid in DNA and RNA and amino acids in protein assembly. So it's the fundamental relation as you're transferring biological information from nucleic acid into proteins. For stringent functional reasons, which I don't have time to go into today, the code must be invariant. That was the standard argument when I was a student. That leads to a strong prediction of a universal genetic code. Now, today in 2021, there are so many variant codes that have been discovered, and there are more discovered every year, that the NCBI here in the United States maintains a web page where if you're working with one of these groups that has a variant code, like Tetrahymena, the ciliated protozoan, or the yeast, Candida, you've got to go look up the code to make sure that you do correct predictions as you read through from your DNA data to predict protein sequence. So the code is no longer universal. Now, when a prediction fails, something has to pay for that. And the question is it going to be the auxiliary theory, or is it going to be common descent? Now, the reason this is problematic is if the auxiliary theory is always paying the price for the failed predictions here at this node, common descent becomes effectively an axiom that cannot be challenged by observation. That is not a healthy state for a science to be in, to have its core theory insulated from empirical challenge. Now, arguably, I won't do this today, we could say that we actually know more about this than we do about this, and that the real arrow here should run from disconfirming evidence to challenge common descent. But keep this logical schema in mind, because it'll be very important when we come to orphans. Big distinction. I'm using...
Sorry, Dr. Paul. Could you just go back one slide? I just have a burning question. So, you know, when you spoke about how you don't just go from common descent to observations, is this related to when Jonathan Wells refers to Ronald Brady in terms of common descent becoming true by definition? If we just don't admit that there's assumptions involved, which are testable, if we just jump directly, it becomes like a definitional truism.
I've just written a book chapter about this where I say that the problem is the formulation of common descent. It's formulated typically in textbooks with generic or common nouns like organism, ancestor, living thing, and all that's postulated is a descent relation where organism A begets organism B, right? And it's at a level of abstraction that you cannot actually take the theory out into nature to generate predictions, because the theory is so generic, it could be true no matter what you observe. So the problem is the theory itself is expressed so generally that it's difficult, or impossible to know in any given case with observation whether the observation is consistent with the theory or not. So the way that you give the theory empirical specificity, where you can actually check a prediction, is you couple it with another part of your biological knowledge that has precision, that has exactitude, and then that lets you make predictions that you can actually check. So you'll see this same pattern over and over in different parts of biology. For instance, in the evolution of the vertebrates, the prediction was made that for functional reasons, development in animals must be conserved because the developmental sequence cannot be perturbed without crashing the embryo. All right, that is something we know from developmental biology. We don't actually know that from the theory of common descent. So if you put that in here and couple that to the common descent now just of the vertebrates, you get a clear prediction that when you look at the embryology of different vertebrate groups, you should see conserved form. In fact, what we see as you move through the different groups just within the vertebrates is a great diversity of early embryogenesis, different cleavage patterns and so forth. But it's the same logic, is that to give common descent empirical specificity, you've got to couple it with other parts of your biological knowledge that are closer to the data themselves. I hope that's helpful. I don't want to spend a lot of time on this because I have so much ground to cover. So a quick distinction that I'm making, the capital letters here refer to the hypothesis put forward by Darwin that all organisms on Earth stem from a single parent. Common descent, small c, small d, you have n+1 parents, okay, where n is to be determined by investigation. The reason for making this distinction is often when I'm in conversation with a biologist, he'll say, how can you possibly doubt common descent? Didn't your parents have parents? Sure, and they had parents and so forth. And what he's doing is trading on an equivocation between these two meanings of that term. So I will always refer to common descent in this talk in terms of big C, big D, and it's the claim of single parent universality. So the domain, as I've indicated, is universal, right? So these little colored boxes here indicate different groups, and there's nothing outside on Earth. This is the universality with respect to life on this planet. If we don't have evidence of LUCA, or we can't form a clear picture of LUCA, it becomes an open question, how many original groups were there? So you can imagine this like a small forest of trees where you're looking at the trees top down as they come out of the earth. So this is a tree, and so forth. And we have 6 here, but we would need more because these groups still haven't been assigned to a lineage. So the geometries will end up being very different if LUCA doesn't exist. Now, common descent is very deeply embedded in my modern biological thinking. When I was a student at Chicago, I read this paper by Francisco Ayala, the geneticist. He says, look, it's as certain as the roundness of the earth or the motion of the planets, right? It's something that you just don't doubt. And you could see this reflected in textbooks. So I used this textbook as an undergraduate. Notice the page number here, right? Page 1, you're entering the discipline of evolutionary biology through the gateway of this claim. It's the first thing you learn. Here's another textbook I used on cladistics by Eldridge and Craycraft. Again, note the page number. This is a cardinal or a primary claim made as you enter the discipline of biological systematics. What's our key proposition? All organisms are related. So I could go on and on about this. It's just taken to be the case, right? So modern biology and all of comparative genetics developed within this framework, going back to Darwin. Here we have Mendel, the rediscovery of Mendelian laws around the turn of the century. This is Dobzhansky, the foundations of neo-Darwinism. Watson and Crick and DNA. This is Frederick Sanger, the time he was sequencing insulin. Zuckerkandl and Pauling, who wrote one of the first papers on using proteomic and genomic data to infer trees of life. Margaret Dayhoff here, who assembled the Dayhoff Atlas of comparative protein structure. All of this knowledge is accumulating within the framework that LUCA was real and the tree of life existed. So when I was a graduate student, I discovered that most academic biologists had very little patience for questions about common descent. So this is a table with a crystal bowl here. These are the topics in science that you can work on successfully and get funded to work on. Then you have the risky topics where the bowl is closer to the edge, okay? You can still work on them. You may have a harder time getting funding. Questioning common descent was down here. You just didn't do it. Now, from the mid-90s until today, this has changed dramatically. So some of you may remember this cover story on New Scientist a little over 10 years ago. Darwin was wrong. Graham Lawton, who writes for New Scientist, was attacked by people like Richard Dawkins. They said he was pandering to creationists, right? Well, if you read the article or you looked at his defense of himself online, Lawton said, no, I'm not doing that at all. I'm simply reporting on what's happening within comparative biology, within the disciplines that deal with the history of life. The work of people, for instance, like Carl Woese. Over the last 2 decades of his career, Woese was increasingly outspoken about the failure of the tree of life picture. In fact, not quite sure where it is. Yeah. This book, The Tangled Tree, recounts this. So Woese is the hero of this book. And Woese became very unhappy with the single tree picture. And in fact, now it's possible to publish on the topic. So Biology Direct is running this series, Beyond the Tree of Life. Very good journal, all open access. I just downloaded this preprint from Biosystems from these 3 authors. Is it possible that cells have had more than one origin? So they're raising the question, maybe common descent's not true, right? It's not even in print yet. But the topic has moved from the periphery, where I dealt with it as a graduate student, right into the center of biology. The question is, if this domain were not real, how would we know? All right. That turns out to be a very difficult question to answer. Now, one would think, well, it deals with every organism on the planet. You should be able to find data everywhere if common descent's not true that would challenge it. Well, it turns out, in terms of the logic of how the theory works, this question ends up being very difficult to answer. But Darwin gave a test. All right, it's in that chapter 6 of The Origin, where he kind of stops his exposition of his theory and takes questions from the audience, so to speak. One of them being, if the theory weren't true, how would you know? Now, it might be he quotes this passage with respect to the mechanism of natural selection, but actually the logic of what Darwin is saying here refers at a deeper level to the natural pathways that must exist from LUCA to any point within the domain of common descent. So think about it this way. By the way, this is not a proper phylogenetic diagram. You could never publish something like this. It's just a cartoon to show this idea of a singularity at LUCA at which all of life is connected. So let's take just one of these segments, A to B. Now you can see, here's LUCA, A. B is some ancestral group. It's still within the domain. So this would not falsify common descent. But we can imagine a point lying outside the domain that would, if it were real, if we observed it. And the reason it would falsify common descent is common descent is universally quantified, right? It has to obtain for every organism on the planet. If there is an organism that cannot be linked by some natural pathway to the rest of life, common descent would be false. Okay, so I'm going to skip that for lack of time. This is known as the principle of continuity, and it's simplicity itself. If you postulate a pathway for the origin of B that commences with A, In other words, that's your phylogenetic pathway, that's your lineage, then every point on that pathway must be biologically possible. Now, that may seem to be a truism. In fact, it's very powerful and widely used within evolutionary theory. This can't happen. You cannot traverse a gulf of biological impossibility, such as an inviable transitional state. So, two important logical notes before we go on. We're almost done laying the logical foundation here. We're almost ready to talk about orphans. The principle of continuity and common descent must be kept independent of each other if continuity is going to act as a judge or referee on any evolutionary hypothesis. Secondly, continuity and common descent entail each other logically. Now, this is critically important because too many biologists think entirely in their imagination that common descent is true, and they never stop to consider what the evidence would be if it weren't true. But if we say that A to B is possible and actually occurred in biological history, by logical symmetry, it might also be the case in light of the evidence that A to B is not possible and did not occur. If this can be true, this can be true. Okay. Now, continuity plays an important role in evolutionary reasoning. So I'll give you just two quick examples to see it in action. Leslie Orgel, in a famous paper in 1968, looking at the origin of the DNA RNA protein system of universally present in terrestrial life, he said, I'm going to be guided by the principle of continuity. And he said, it's very hard to see how a different chemistry that is different from the one that we know could have given rise to the one that we observe. So here we have DNA, RNA, protein. This is what we have in our biochemistry and cell biology textbooks. And Orgel is saying, if you start with a fundamentally different chemistry that in particular is storing information, at some point, you're going to have to transfer all that information over to a very different chemistry, maintaining viability all along this pathway. He says, I see no way that that could happen. So he uses continuity to rule out these kinds of hypotheses, which were present in the literature at the time, and still a few people talking about them today. I'm going to skip this example because I'm looking at the clock and I've got so much ground to cover. Just another example of the principle of continuity. All right, logical problem. How can anything lie outside a universal domain? Well, what Darwin did and his intellectual offspring have done is to use what are known as counterfactual conditionals, where they say, if it were the case that X, right, where X is going to be filled in by some counterfactual observation, then we would know that common descent is false. So we should not observe a lot of unique genes and proteins. This is what orphans are going to represent when we come to them. We shouldn't observe variant genetic codes. We shouldn't observe incongruent phylogenies, where the tree built with morphology is not congruent with the tree built from molecular data. We shouldn't observe fossils out of stratigraphic sequence and so forth. Now, what's happened in this case is the domain of common descent has swelled, excuse me, swelled to take these in. This one, this one.
Do you want to take a break for water while I... So guys, just to, in other words, for anyone who's just joined us, the principle of continuity has to be separate to common descent. It can't be the same thing. It's completely untestable. And, like Paul has said, this is something that biologists don't even think about. What would be the observations if common ancestry was actually false? It's not a possibility for them.
Yes, thank you very much. Although I think increasingly people are thinking about these questions because of the genomic data we're going to look at. So let's look at unique genes and proteins. One would think that a theory that obtains over a universal domain would have searched most of that domain. In fact, in the case of common descent, only a tiny portion has been searched. And what I mean by that is prior to the invention of automated DNA sequencing and it becoming a practical technology in the mid-1990s, no one had a clue how diverse the genetic universe was on planet Earth. And you can see that in this figure. This shows the growth of deposition of sequence data, both bases in the blue and individual sequences in the orange line here, up to November 2016. And I need to update this figure to February 2021, but it's the one that I have at the moment. I was an undergraduate studying evolutionary biology back here. My graduate career spanned 1985 to 1997. Most of what we know about the genetics of life on this planet was learned in the past 25 years. A curve like this tells you that the generalizations that you drew over here are overwhelmingly likely to be wrong because you just didn't know enough. Okay, and of course now both of these curves are way up off the top of the screen. So we can lay that curve down here and say, we put in biological theory going back to Darwin, right up until the structure of DNA. In fact, right up until 1995, when automated sequencing became a practical technology, we just didn't know. And that tells you that the theoretical assumptions that you are making prior to this huge influx of data, you really can't blame yourself, but they are likely to have been wrong. So enter the orphans. Now, in the mid-90s, the first eukaryote to undergo complete genome sequencing was yeast. And as the yeast sequence came in, Bernard Dujon and others at the Institut Pasteur noticed that what they call the mystery of orphans just jumped out at them from the sequence. And they put it in this review paper. Approximately half of the protein coding open reading frames had no clear-cut sequence homologs in any organism, including yeast itself. So in this review article, he said, what surprised us? A significant portion or proportion of yeast genes are orphans of unpredictable function. Okay, so I believe it was Dujon who coined the term orphan gene. And there's a puzzle, at least, or not a puzzle, there's a pun in the name. In molecular biology, ORF stands for open reading frame. So this is a typical prokaryotic protein coding sequence where you have a long so-called reading frame where there are no stops. No stop codons, unless they're specially translated by the species that have that apparatus. And during translation, an amino acid is inserted at an in-frame stop, but I won't consider that exception here. So this sequence will have characteristic features that you can write algorithms to detect. For instance, there'll be a promoter region upstream here, a start codon. Again, a long sequence of codons, 4 amino acids with no stops that are in frame, and then a stop codon. And the sequence, and these features are so general that you can write algorithms to look for them. And then you take your newly sequenced DNA and you feed it into GenBank or any other database where annotated sequences are stored, looking for base pair by base pair hits using matching algorithms like BLAST. So here we have, in this cartoon, we have 4 sequences. We submit our newly sequenced DNA and we get a hit. In other words, the matching algorithm is able to assign a high degree of similarity between an existing annotated sequence and the one that we just obtained, and now we're looking for its molecular kin, right? A good parallel to this, by the way, comes from natural language. I've got a book here. I see a word that I don't know, so I go to my Oxford English Dictionary, which is over there, and I look for the corresponding character string. So the already deposited sequence is coding for a ribosomal protein. We get a hit. That tells us what our new sequence is doing. With orphans, that doesn't happen. We deposit or we submit the orphan sequence to BLAST, and we get back a report saying no significant similarity found. Okay. Now, remember, we have good reason to believe our newly sequenced piece of DNA codes for something because it has the features of a protein coding sequence, but the matching algorithm we're using does not find any corresponding match. Next time on the 15th, I will talk about new work done by Kara Weissman at Harvard and others to say that these matching algorithms are defective. In other words, sequences can diverge into a twilight zone beyond which they can be detected. And I'll be looking in detail at her analysis, but that's for part 2. So Russell Doolittle, who spent his career working on these questions, in a review paper in Nature in 2002, so probably I think a few dozen genomes were available at the time, he said, it's the biggest surprise. In the genomes that we've looked at so far, we find vast numbers of putative genes for proteins of unknown function. And we don't know what they're doing or where they came from. So he's looking at the signal right about here. And of course, it's very much stronger now. He goes on, how could there be so much in the way of unknown equipment? Large numbers of unidentified genes in a variety of organisms that look conventional in every way. Where these unique sequences are coming from and what they do remain baffling mysteries. Now, here's a distinction that I failed to make several years ago in a talk in Florida, and someone wrote up the talk for the site Panda's Thumb and really laid into me for the failure. And it hurt so bad. Making this mistake was so painful that I vowed never to give a talk on this subject without making this distinction. So you're benefiting from my failure, okay? This is a Venn diagram. Genes of unknown function is the larger class. Orphans nest within that. So you can have genes of unknown function that are present in multiple species. It's the same sequence, but because they are present in multiple species, they are, by the strictest definition, not orphans. So the mnemonic is all orphans are genes of unknown function, at least when first discovered. The opposite is not true. Okay? Keep that in mind. Now, Richard Buggs and I, he's a botanist, just published a really interesting paper on the origin of the angiosperms. He and I wrote a chapter for this book called Next Generation Systematics, where the editors in their introduction said, Nelson and Buggs have identified a really interesting puzzle, the increasing presence of orphan or taxonomically restricted genes. They are expanding in number, and they appear not to be junk, right? They appear to be functional, at least when they're investigated. So Richard, as you may know, sequenced the genome of the ash. He works at Kew, Royal Botanic Gardens, on diseases of plants. And ash suffer from a disease known generically as ash dieback. But some species of the ash are resistant. So he sequenced the ash genome in multiple groups to try to find the molecular correspondence that enabled resistance. And then he wrote up a review, or not a review, a blog entry for Nature, Ecology, and Evolution, because approximately 25% of the ash genome was orphans. So he wrote this article about it, and he continues to maintain an interest in this. So here's the flow chart. You've got your new data, your new sequence data. Taxonomic category will be species or genus. You will submit that to the NCBI or the EMBL database, non-redundant database. So that's your dictionary, the second node there. And you will use a homology threshold. So this is an adjustable parameter in BLAST that you can tweak. And as you make it more stringent, you will have a better estimate of a reliable hit from within the genomic database. That pipeline then will give you your orphans. These are the protein. I'm talking today, by the way, only about protein coding sequences, not sequences coding for functional RNAs. So strictly protein coding sequences. That pipeline will then give you your orphans. So starting here with your raw data and analytically working this way, and then these are your candidate orphans. So when orphans were first discovered, didn't take very many genomes for the pattern to jump out at people. They caused some head scratching, right? Where were their necessary intermediates? This Israeli team in a paper in 2003 said, we didn't expect to see them. Why? Why were they surprised? Well, Russell Doolittle at UC San Diego wrote another review paper in 2005. He said, we thought that we could identify most genes just in silico, because extant genes would be descended from a smaller ancestral population that expanded by gene duplication. So in this cartoon, you have of course, real gene would be very much longer than this. You're coding for, your DNA is coding for a protein that looks like this. That sequence undergoes a variety of evolutionary processes, and you add an alpha helix here. But the backbone of these two proteins is going to be largely the same, except for this additional structure here. These would show up as having the same fold, more or less, right? Be classified together. And these genes will carry a signal of history. It won't be entirely wiped out. So for any gene or protein in the tree of life, one ought to be able, using basically similarity matching, to work your way back down these lineages to the minimal set that was present in LUCA. So in the background of Doolittle's mind and the background of the Israeli researchers' minds is common descent is a given. That's not up for grabs. New genes and proteins come from old by well understood processes. If one is the case, and we understand two, the last thing we would expect to find are lots of unknown genes and proteins. That's why orphans were so surprising, because the theoretical background did not predict them. And I'm going to move quickly through these slides so we can get to the data. But what these slides indicate is nobody expected orphans. I lived through this, so I saw it firsthand. As a student, both as an undergraduate and a graduate student, orphans were not on anyone's radar screen. They really shocked people. So these slides are summarizing a very large literature where the investigators are saying, we didn't expect this, okay? And I'm going to skip that. I apologize. I have so much material, but I want to stay on track. I will take a moment to talk about this. This is Aoife McSlight, or McClysick. That's hard to pronounce her Irish name. I apologize for that. She is a leading investigator on orphans. And in this paper, she points out that in the early 1990s, prior to genomic sequencing, de novo origin was considered so improbable as to be impossible for more recent evolution, right? Nobody expected it. It goes on in the same paper. It has now become clear that de novo origin is a consistent feature of eukaryotic genomes. Yeast, plants, flies, mammals, primates, even human evolution. What happened was the wind changed direction 180 degrees. Now, that happens in science. New data surprise people and they change their theoretical expectations. The problem is, if we go back to our logic schema, we need to remember what it was that we originally predicted. Common descent plus a strong theory about the low probability of the formation of genes and proteins de novo gave us the expectation few, if any, orphan genes and proteins. When that turns out not to be true, somebody over here has got to pay. And the question is it going to be the auxiliary theory or is it going to be common descent? And again, I want to stress this point. If you let the blow be absorbed all the time here, you render this effectively an axiom that cannot be challenged by observation. And that is not a healthy position for any science to be in. Subor?
Yeah.
I am looking at 4 minutes remaining.
That's fine. We can continue this another time.
All right. Why don't I do this? Why don't I finish it too? Most of the interesting biology is yet to come. I spent so much time laying out the logic that all the cool biology is in the second half of the talk. And what I would like to do is, rather than rush as I've been doing and dropping slides and so forth, finish up with this transposon example and then reschedule another session when I can, because I have probably 100 more slides to go.
Yeah, I mean, one thing I would suggest that we do, Paul, is that we could do it not to cover it per session, but just leave it as a series until you've covered it thoroughly. Because there's questions I had, but I just held back because of time. So whenever you're available, every 6 weeks, 7 weeks, whenever it is, we can set this up.
All right. Well, why don't we do this then? We can stop here, and we'll. I'm sorry if I disappointed anyone who's watching.
No. I mean, there was enough for people to digest. I think more than enough.
Right. When I was growing up, this probably may have been the case in your family. Dessert, the pudding was always served at the end of the meal, right? So the pudding is yet to come, right? It's all the interesting, cool biology. So I would love to be able to finish this, but maybe we should take questions now.
Yeah. What we would do is, firstly, I hope everybody's understood the framework, because the next session and session up may take actually 2 sessions rather than 1. What we're going to be doing is looking at not only the interesting biology when it comes to the data that's coming in terms of the orphan genes, but also the contentions which are put against the idea that this challenges common descent, by saying, oh, de novo, genes coming together, that's just to be expected, right? So I think I want to first ask you a question here, a sort of Kuhnian question. I mean, this seems like almost a perfect example of normal science, and people resisting changing the paradigm and actually just shoehorning the data to just reinforce common descent, because de novo, like I think the quotation that you had was from the early 2000s or the 1990s, what was ruled out. It was not something even someone spoke about. And like you said, orphan genes was not something that, was even predicted. So how much of that, apart from the science, the philosophy of science, because you've studied obviously, and you've published on the philosophy of biology, like, I want to hear that perspective in regards to this.
Yeah, it's entirely reasonable to protect your core commitments. Every science does this, right? Imre Lakatos had a model of scientific inference where great philosopher of science, unfortunately no longer with us, but his model was that you had a core set of theoretical commitments, and around that, a kind of belt of secondary commitments or auxiliary commitments that you didn't hold as tightly. And that when, as is always the case with any scientific theory, nature talks back to you. You find disconfirming data, you find anomalies, that the way that they're absorbed by your theoretical bundle is differentiated by the relative degree of strength you assign to your core versus your belt of auxiliary. So in one sense, absorbing, if I can throw up one slide here.
It's kind of like a web of beliefs, where it's like a view of foundationalism in philosophy, where the strongest beliefs are at the core and everything you're willing to give up is as you expand out, right?
That's right. And so I think the history of science shows this very clearly. So I'm not faulting evolutionary biologists for doing what comes naturally to any scientist. What I'm doing is sounding a note of caution about this practice as the strength of the anomaly grows.
Yeah.
Right? And recognizing that when a prediction fails and you have to pay the cost for that, you as investigator have a choice to make. Where are you going to focus your critical analysis? And the problem is, if it's always being absorbed here, this ends up not telling you anything, because the specificity of common descent, its ability to actually make predictions about nature, is obtained in companion or in tandem with its auxiliary theories. And this ends up becoming an axiom. Now, axioms are wonderful in geometry. They're wonderful in the exact sciences of logic and mathematics. Axioms are bad news in empirical science.
Yep.
Where there needs to be traffic, if I can use this metaphor, between nature and our theories, where it's a two-lane highway. The traffic is going both ways. So I personally have lived through the failure of major evolutionary predictions, such as I mentioned earlier, the universality of the genetic code. It troubled me very deeply when that prediction failed, that no one took it seriously. Well, the code can evolve. Who knew? All right, you can do that, but your core theory is going to pay a cost, and you will end up bleeding it white of its content.
Absolutely. And just the last thing I'd like to put to you, because I know you have to go now, and thank you for giving the information that you did today. And I you spoke about dessert being at the end, but I think people were enjoying it throughout. So obviously you've sympathized with the other side. That's fine. But let's just go back to when we faced a similar issue in regards to, okay, so Mercury is wobbling. There must be some planet which has a gravitational pull. So how many of these types of Vulcans are we going to have to see before someone actually says, wait a minute, because or else in a way, Paul, I mean, the great stuff that you've done and the risky prediction that you made and all this stuff, the data seems, I mean, the anomalies seem like, okay, they're just accumulating, but at what point do you cut off, and how do you foresee that coming?
I think it's happening already. So one of the parts of the pudding, actually, there were several puddings on the side table waiting to be served. One of them was, for reasons entirely unrelated to the intelligent design controversy, major investigators are bailing out of the single tree picture. So at an accelerating rate. And they are saying it's no longer possible to reconcile the data we have with this singularity of LUCA. And a variety of proposals are on the table, such as, instead of a bifurcating tree, Steiner tree with a single root, a network where you have just this Roman orgy of horizontal gene transfer from multiple independent starting points. There's just a panorama of different models on the table now within evolutionary theory, again, unrelated to the intelligent design controversy. So already, I think the process of, as this diagram shows, of rethinking common descent is underway. Both in the second half of this talk, which I didn't get to, but much more on the 15th, when I present again to a UK audience, I'll be looking at some of these proposals, if I can get to them, of, what if there were multiple independent starting points? What would we see? So the anomalies are so dramatic, I think, now, not just in orphan data, but actually in key features of living things, such as the ribosome. When I was a student, ribosomes were illustrated as a kind of cartoon with the large subunit, the small subunit, and pretty much any ribosome is the same as any other ribosome. We now know that is no longer the case. Ribosomes are incredibly diverse. Yes, they're all doing the same thing, but almost on a species level, the molecular disparities present in that key molecular machine are very striking. Can that be reconciled with a single origin story? I don't believe so. So I think the next decade is going to be incredibly interesting. And one final point, if you take a census of nucleic acid on this planet, most of it is in the virosphere. So in a single liter of seawater, there are billions of viruses, and the molecular diversity of viruses will blow your mind. all of that nucleic acid has to find an origin. And 4 billion years seems like a long time, but when you look at the actual diversity of the virosphere, it's astonishing. And if an alien biologist came from Alpha Centauri and landed here and started doing a census of all the nucleic acid present, he would say, well, cells are one thing. Where did all this nucleic acid come from in the viruses?
Yeah.
I'll try to talk about that next time.
Absolutely.
It's an exciting time in biology. I tell students, forget about physics. They can't build accelerators big enough to test their theories, right? If you want a science that's on fire with interesting data, biology is your game.
Absolutely. Thank you so much, Paul, for your time. We had lots of people really appreciating, your thoughts, your perspectives. And in a way, this is like an exclusive thing because this information is like very hard to get. I mean, this is not the standard narrative, of course, and this is the cutting edge counter narrative. So thank you very much, Paul. Hope to have you soon again. And next time we will interact with the audience a lot more. For everybody watching, share this video like last time, tell your friends about it, spread it around, and hopefully we are going to have Dr. Paul Nelson back again in a few weeks' time, speaking about this very important topic.
Thank you. I look forward to joining you